Then we will learn about the M phase, and in this we will learn about the karyokinesis first, which is the first phase of M phase. So what do we mean by karyokinesis? The karyokinesis is a term which we use for the division of nucleus. Karyon means nucleus, and the division of the nucleus is called as karyokinesis. So before the cell division takes place, cell divides its nucleus. So nucleus should be doubled before the cell division, and then cytokinesis is done. So the cytokinesis means the division of the cytoplasm. Once we cell is after completion of cell division, the nucleus division, cell will divide its cytoplasm to produce new daughter cells. Okay, so first phase of this karyokinesis is prophase. So let me just erase the all stuff about interphase, then we will start. Please wait. So let's continue from the karyokinesis of the mitosis. So firstly, we will learn about the mitosis. What is mitosis? Mitosis is a type of cell division in which two daughter cells are produced from one single cell. Okay, so let's say, suppose this is a parent cell. Suppose this is diploid cell, which means it will have two sets of chromosomes, right? So what will happen? The cell will divide to form two cells at the end of the mitosis. We will have two cells, and those cells will have the diploid nucleus, which means the chromosomes are not getting reduced. Diploid cell is producing diploid daughter cells, but the DNA content is reduced. That's the thing we have to notice. The DNA is reduced, but not the chromosomes. Okay, so this one thing you should know about the mitosis. The diploid cell will produce diploid cells. One cell will divide into two cells. Okay, in the first phase of mitosis, it's called as karyokinesis. So let's start from the karyokinesis, which is the division of nucleus. Division of nucleus. Let me just erase this. So karyokinesis is again divided into four phases: prophase, metaphase, anaphase, and telophase. You can remember the sequence of these phases with the help of this mnemonic PMAT. Prophase, metaphase, anaphase, and telophase. So just we have to remember what happens in the prophase, what happens in the metaphase, what happens in the anaphase, and what happens in the telophase, and as well as the diagram of these phases. So let's learn about the prophase first. What will happen in the prophase? Cell is fully matured, and cell will start. In the cell, you will start to see the disappearance of the nuclear membrane. So this is the first thing happens in the prophase. Nuclear membrane starts getting disappeared. The second thing you will see, the chromatin condense to form chromosomes. So condensation of chromatin takes place. Two important things you have to remember. Two important events. First is the disappearance of the nuclear membrane. nuclear membrane disappears. Second, the condensation of chromosomes or chromatin, you can say chromosomes. So chromatin is getting condensed, then we can see the chromosomes. Because normally in the interphase, the DNA is in the form of chromatin, which is a loosed form of the DNA. But if you condense and compact that chromatin, the chromosomes are visible. So condensation of chromatin takes place in the prophase. Then after the completion of prophase, cell enters into the M phase. What happens in the M phase? Sorry, my bad. Metaphase, not M phase. Metaphase. So two important things again. All the chromosomes lie on the equator of the cell. This is the equator of the cell, and the chromosome will lie like this. And these centrioles will move towards the opposite poles of the cell. Centrioles, which has very important role in the cell division, they are going to synthesize the spindle fibers and spindle apparatus for the cell division. So they will move to the opposite poles of the cell, and then the chromosomes will lie at the equator of the cell. And what happens in the metaphase? Centrioles. These centrioles will start forming spindle fibers as well as asters. Asters are the fibers which hold these centrioles, and the spindle fibers are the fibers which helps to separate these sister chromatids from each other. So these spindle fibers will be formed and they will be attached to the kinetochore, which is a disc-like structure present on the centromere of the chromosome, and these spindle fibers will be attached to the kinetochore. Okay, so these are the events takes place in the metaphase. The chromosomes are lying on the equator and forming the metaphase plate. This is known as metaphase plate. The centrioles moves to the opposite poles of the cell. They form spindle apparatus, asters and spindle fibers, and they will attach to the kinetochore of the centromere. Right? So now these sister chromatids are ready to split and get pulled to the opposite poles of this.